Good morning, welcome to VSPG, everyone. Today we have a talk by Dr. Lidya Tarhan from Yale University titled Life and Death in the Ediacaran, Evolutionary, Environmental, and Preservational Dynamics. Lidya will talk for about 45 minutes, and then we'll transition to some Q&A. And after the Q&A, we can open to a more open discussion style. Next week, we have a presentation, again, by Elizabeth Turner from Laurentian University. We had someone drop out of the April 14th spot on super short notice, and Elizabeth jumped in really quick, so we're really thankful. That talk is titled, Exploring the Timing of Mineralization in the Giant Neoproterozoic-Hosted Kamoa-Kukala copper deposits, Central African Copper Belt, Democratic Republic of Congo. So that's next week. And with that, Andre, do you have any announcements? No? Okay.
We finishing schedule up to New Year. So if you're curious, you can probably go and see. I think Alex already posted.
Oh, it's not posted yet. I was going to post it once it was final.
And starting with November, December, we will switch to a new mode. So we give a chance for people from Australia, Asia to talk, and we will do presentation in afternoon, I think at 5 o'clock.
Yeah, 5 o'clock Pacific. So we'll make more announcements for that soon. Okay, I'll introduce Lidya for us. So Lidya Tarhan's research focuses on using the sedimentary record to reconstruct the coevolution of ancient life and environments during critical intervals of Earth history. She also investigates the processes responsible for fossilization and the manner in which preservational biases modify geologic archives. Her research approach combines field-based paleontological and sedimentological investigations with a petrographic geochemical and modeling toolkit. She has done fieldwork worldwide with a particular focus on Western USA and the Flinders Ranges region in South Australia. Her recent work is centered on reconstructing the radiation of multicellular life, as well as the emergence of early animals as ecosystem engineers. In addition to her geologic investigations, she also studies the environmental factors shaping animal-sediment interactions in modern marine sediments. Thank you so much, Lidya, for being willing to speak for us, and please take it away.
Thanks very much, Alex, for that kind introduction, and thanks to both you and Andre for the invitation to speak in this seminar. I'm really delighted to have the opportunity today to share some of the recent work that my group has been doing on constraining some of the more dynamic aspects of the Ediacaran seafloor. So the Ediacara Biota is Earth's earliest fossil record of communities of complex, macroscopic, multicellular organisms. And Ediacara fossils occur around the world in assemblages of Upper Ediacaran age, predominantly in what we call the Ediacara-style, in which these entirely soft-bodied organisms are preserved three-dimensionally as casts and molds and sandstones. And although as a community, we've been chipping it away at a number of important questions regarding the Ediacara biota, including the morphologies, the ecologies, and even to some extent the affinities of individual Ediacara taxa, I would argue that there are still a number of broader scale questions regarding the Ediacara biota and its place in the emergence of complex life that remain under-addressed. Such as what factors control the distribution of Ediacara fossil taxa in time and space? How do we go about reconstructing the structure of Ediacara seafloor communities? And can we reliably track changes, turnovers, and taxonomic diversity within the Ediacaran? And even to what extent might the Ediacara fossil record be biased, or alternatively, provide us with a fairly faithful reflection of life and death on the Ediacaran seafloor? And in order to address any of these questions, what we really need is a field-based approach. We need to be able to characterize Ediacara fossils within their sedimentological and preservational context in order to determine to what extent they vary at the specimen, at the bedding plane, at the facies, and the assemblage scale. And perhaps even more crucially, we need to resolve what sorts of processes, what sorts of mechanisms were responsible for Ediacara-style preservation, for this mode of fossilization that is so disproportionately responsible for our windows into Ediacaran life and fossilization. And perhaps one of the best places that we can go in order to interrogate some of these questions of Ediacaran environments, Ediacara ecology, and fossilization is the Ediacaran of South Australia, and in particular, the eponymous Ediacara member of the Ronsley quartzite, which crops out throughout the Flinders Ranges region, and in which taxonomically rich assemblages of Ediacara biota fossils occur in a variety of sandstone-dominated shallow marine facies. Moreover, at sites such as Nilpena, here on the western margin of the Flinders, which recently became an Ediacara National Park, ongoing excavation and reconstruction of fossiliferous bedding plains has provided us with an unusually highly resolved window into some of the more dynamic aspects of the Ediacaran seafloor. And fossil assemblages in the Ediacara biota, much like many fossil assemblages in other Ediacara biota deposits, are characterized by this distinctive Ediacara style of preservation, in which these soft-bodied organisms are preserved three-dimensionally as casts and molds or impressions in sandstones. However, the mechanisms responsible for this distinctive mode of Ediacara fossilization have long remained somewhat more elusive. Some have postulated that rapid burial, that abrution of these seafloor communities by underwater storm events may have played a pivotal role in Ediacara-style fossilization. However, rapid burial is certainly not unique to the Ediacaran. So although it may have been an important contributing factor, as it was for a wide range of other Phanerozoic and Precambrian modes of exceptional fossilization, it's unlikely to have been the principal driver of Ediacara-style fossilization. Alternatively, some have suggested that the tissue consistency of Ediacara organisms themselves, and in particular, how resistant or how prone to degradation or collapse they were, may have played a pivotal role in driving this distinctive modic style of preservation. However, as recent work has very nicely highlighted, Ediacara-style preservation is not synonymous with Ediacara biota taxa. And in fact, some of the same species, some of the same organisms that we see preserved moldically in this distinctive Ediacara style in units like the Ediacara member have also been characterized in very different and more familiar modes of fossilization, such as preservation as thin carbonaceous compressions, or what in the Phanerozoic we would often call Burgess shale type preservation in other Ediacaran aged units, such as the Doshan 12 Formation of South China. So tissue consistency, much like rapid burial, is unlikely to be the principal driver of Ediacara-style fossilization. Moreover, Ediacara-style fossilization is not limited to either the Ediacara biota or the Ediacaran period. And in fact, we have examples of this distinctive style of fossilization that extend as far back as the Mesoproterozoic. And in fact, some of our earliest candidate multicellular eukaryotic organisms, such as Horodischia, are preserved in this moldic style. And we have examples of Ediacara-style fossilization that extend to as recently as the close of the Lower Paleozoic, in which very different sorts of organisms, definitive crown group animals, such as various groups of arthropods and Scyphozoa and Medusae or jellyfish, are preserved moldically in this same Ediacara style. And this would suggest that perhaps when we're searching for mechanisms of Ediacara-style preservation, we should be thinking not only of global scale and environmental drivers of fossilization, but also perhaps drivers that could have operated over a much broader swath of Earth's history than has historically been appreciated. Sorry, I just lost my forward button here. All right, and when we characterize Ediacara-style fossil assemblages in detail at the bedding plane scale, as facilitated by sites of ongoing excavation reconstruction, such as Nilpena, one of the things that most readily becomes apparent is that Ediacara-style fossilization is not unique to our more charismatic macrofossils, but also characterizes some of the smaller and cellular components of the Ediacaran seafloor, such as organic or microbial macro. macgrounds, as preserved by features like textured organic surfaces, some examples of which you see here. And these textured organic surfaces provide us with an important first order line of evidence for the presence, in some cases, the pervasive presence of organic or microbial mats on the Ediacaran seafloor and the manner in which these macgrounds were shaped by hydrodynamic and sedimentary processes. Moreover, these textured organic surfaces also indicate that although Ediacaran mats likely included consortia of microbial grade organisms, such as bacteria and archaea, much like mats today, they also, and rather more unusually, included dense aggregations of macroscopic and multicellular grade organisms, such as dense aggregations of tubular fossils, such as this tubular fossil, Phoenicia. And it is not only the presence of features like these textured organic surfaces, but also the record of the manner in which Ediacara macroorganisms interacted with the microbially or organically bound seafloor sediments in the form of the trace fossil record that tell us that many Ediacara Biota organisms were particularly adapted to a mat ground way of life, that they practiced very specific mat ground ecologies. So for example, many of these organisms appear to have utilized the organic macground as a source of nutrition. So the fan-shaped arrays that we term Kimbericnus, the trace fossil Kimbericnus, that are characteristically associated with the Ediacara macrofossil Kimberella are one of the principal lines of evidence that have led to the suggestion that Kimberella may have practiced a mat scratching or rasping or grazing feeding ecology, analogous to that practiced by many modern groups of mollusks such as gastropods. Other Ediacara organisms appear to have utilized the buried interfaces of microbial macgrounds as a source of nutritional matter, as indicated by some of the phenomenal undermat mining trace fossils, such as this one, Helminthoidicnites, that we see in upper Ediacaran successions. And other organisms appear to have utilized the organic macground as a means of stabilizing themselves in the Ediacaran substrate, which was likely important in high energy environments, such as those recorded by the Ediacara member that would have otherwise been characterized by shifting sands. And these include not only some of our tubular macrofossils, but also some of our more iconic frondose organisms, such as Arborea. And these evidences for macground adaptations in the form of these trace fossils, as well as textured organic surfaces and the morphology of Ediacara organisms themselves, are important not only in providing a window into the ecology of the Ediacara biota, but also because they record a number of important firsts on the road towards the emergence of complex animal life. The earliest evidence of movement, the earliest evidence of burrowing, even some of our earliest evidence for biomineralization appear here in the Ediacaran and appear to have originated as mat ground adaptations. Now, insights into the manner in which the microbially or organically bound substrate shaped the Ediacaran seafloor come not only from textured organic surfaces and trace fossils, but also from erosive sedimentary structures, such as these sandstone-hosted sandstone intraclass, in which we've interpreted to be microbial mat rip-ups. And in fact, some of these intraclass still contain the vestiges of textured organic surfaces. And these sandstone-hosted sandstone intraclass are distinctive for a couple of reasons. One is that they record brittle deformation. In other words, in order to be ripped up, in order to be created and resedimented and redeposited in this manner, they must have been derived from a substrate that underwent early lithification. The other distinctive feature about these intraclass is that they are hosted within a sandstone matrix that is texturally and compositionally indistinguishable from that of the class themselves. Yet they're nonetheless preserved as discrete sedimentary objects on the upper surfaces of these sandstone event beds. So a rather unusual style of sedimentation, and also perhaps telling us something unique about the diagenetic history of Ediacaran seafloor sediments. And this is something that I'll come back to later on in the talk. But to circle back to this question of Ediacara-style preservation, what mechanisms, what processes could feasibly offer some explanation for this distinctive moldic style of preservation? And the hypothesis that perhaps has received the greatest attention over the past few decades is the death mask model, originally proposed by my colleague Jim Gehling. And Jim suggested that the presence of macgrounds on the Ediacara seafloor was important for not only the ecology of Ediacara organisms, but may have played a direct role in shaping their fossilization. And in particular, the activities of sulfate-reducing microbes within these mat grounds may have facilitated the precipitation of a pyrite veneer or crust along the interfaces of these buried communities, and that pyritic crust or death mass in turn facilitated this distinctive moldic preservation. And the death mass model was really seminal in that it brought renewed attention to the very unusual fossilization of these deposits, and that it also sought a mechanistic explanation for Ediacara style for moldic preservation. However, in the ensuing decades since the death mass model was first proposed, additional observations, including the sedimentology of Ediacaran fossiliferous deposits at the field scale, the specimen scale, and the petrographic scale, as well as the emergence or application of emerging geochemical techniques, have suggested that the rusty or hematitic iron oxide-rich coatings, which are pervasive on many Ediacaran fossiliferous bedding planes in units such as the Ediacara member, and which have been mooted by Jim as the oxidized remnants of pyritic death mass, can in fact be much more parsimoniously interpreted as evidence of recent alteration of groundwater source precipitates that formed within at most the past couple of millions of years. And when we characterize these hematite-rich coatings in the field, what we see is that they are not bed penetrative, that they are confined to the upper and lower surfaces of bedding planes, and they often have very sharp junctions, whether it's between ripple crests and croft, whether it's within individual fossils, or commonly between contiguous pieces of bedding plane that were subsequently reconstructed but excavated from different areas of the hill slope. And we can also apply geochemical tools, such as the uranium isotope system, to directly test whether these iron oxides record evidence for recent diagenetic alteration and geochemically open system behavior. And in particular, we can use the ratio of uranium-234 to uranium-238, which is commonly termed the activity ratio. And this is because uranium is commonly a co-precipitate under oxidizing conditions with iron oxides. And of course, both of these isotopes are part of a larger radiogenic decay series, whereby over long geologic timescales, uranium-238 undergoes alpha decay to thorium-234, which subsequently, and on much shorter timescales via a couple of intermediaries, undergoes beta decay, producing uranium-234. And due to alpha recoil and damage incurred to the mineral lattice during this process, uranium-234 can then, under oxidizing conditions, be preferentially leached to solution. And once it is in solution, it is a common co-precipitate, again, under oxidizing conditions with iron oxides. And the uranium activity ratio is a proxy that's commonly been used to determine the age and the uranium content of aquifers. However, importantly for our purposes, it's also a tool that can be used to test for recent diagenetic alteration and recent open system behavior within oxides associated with Ediacaran fossiliferous surfaces. And this is because regardless of your starting composition, your initial activity ratio, on relatively rapid timescales, if we have a geochemically closed system, this activity ratio will re-equilibrate to a value of 1, which we call secular equilibrium. So if we were to observe in oxides associated with Ediacara fossils, not only autogenic uranium enrichments, but enrichments which are associated with activity ratios of any other value than 1, we would know that we are at secular disequilibrium, and this would be an unambiguous indicator of open system behavior within the past couple of millions of years. And what we observe is that when we characterize the oxides associated with fossils in the Ediacara member across taxa, across bedding planes, across facies, they are commonly associated with autogenic uranium enrichments elevated above crustal values, and all of these enrichments are characterized by activity ratios which are not at secular equilibrium, and thus indicate that these oxides are characterized by geochemically open system behavior and likely formed within the past couple of millions of years due to interactions with oxidizing groundwaters, but more fundamentally, based on these data, we cannot use the presence or the distribution of these oxides to say anything about Ediacaran iron or sulfur cycling or fossilization. And from a more mechanistic perspective, pyrite and its precursor phases, such as iron monosulfides, are very common components of marine sediments today. And sulfate-reducing bacteria have, of course, been around for billions of years in a variety of surface environments. But pyrite and iron monosulfates don't tend to be strong cementing agents. They tend to precipitate as isolated spherules, and they certainly don't tend to lead to pyrite cementation of sandstones. In other words, to understand Ediacara-style preservation, what we really need to explain is how is it that the sand grains of which these fossils, of which these casts and molds are comprised, became cemented together in the first place? And how did that happen rapidly enough to capture all of this phenomenal morphological and anatomical detail? So a few years ago, casting around for potential alternative explanations for Ediacara-style preservation, I was struck by the temporal correspondence by this interval in the sedimentary record in which we see Ediacara-style fossil deposits, the Proterozoic through the lower Paleozoic, and independent indicators from geochemical, from sedimentary and paleontological proxies, indicating that the dissolved silica levels of Earth's earlier oceans were much higher than they are today, potentially as high as at saturation with amorphous silica or cristobalide. Today, in contrast, the oceans are largely depauperate in dissolved silica, and this is due to continual uptake, continual drawdown by silica biomineralizing organisms, such as silicifying sponges and radiolarians and diatoms. But could the much higher dissolved silica levels characteristic of Earth's past oceans potentially offer some explanation for Ediacara-style preservation? And as we initially described a few years ago, Ediacara-style fossils at first glance, such as those of the Ediacara member, do indeed appear to be very silica-rich rocks. In the Ediacara member, these are typically quartz-cemented quartz aeronites, in which petrographic characterization, as well as electron microscope characterization, so here we're looking at some secondary electron images, as well as the cathodoluminescence maps, these quartz cements occlude nearly the entire porosity of these rocks to the exclusion of all other phases and occur as syntaxial overgrowths around the detrital quartz and feldspar grains. So from a textural perspective, especially since we see very limited evidence for dissolution and reprecipitation, these seem to have formed at a relatively early pre-compactional stage in the burial history of these sediments. Moreover, we can also characterize the trace element composition of these Ediacara fossil-associated quartz cements and compare it to that of the surrounding detrital quartz grains. And for instance, we can measure the ratio of germanium to silicon in these cements using laser ablation inductively coupled plasma source mass spectrometry. And what we see, as you can see from this overlay of laser ablation data upon this cathodoluminescence map, is that the cements are characteristically associated with different germanium silicon values relative to that of the surrounding detrital quartz grains with which they are otherwise mineralogically equivalent. And use of the germanium silicon proxy is important in this application because it allows us to determine whether or not these cements have grain-like signatures. In other words, could they feasibly have been formed by high pressure or high temperature pathways of remobilization of the silica in those detrital grains, such as we would typically envision to be pathways of quartzite formation, or did they have an exogenous origin? And this signal that we see for higher germanium silicon ratios in the cements relative to the grains is something that we see at a much broader scale across the Ediacara member, across taxa, across bedding plane assemblages, across facies. Characteristically, Ediacara fossil cements have different and slightly higher germanium silicon values than what we see in the detrital quartz grains. And that indicates that thermodynamically speaking, they could not have formed through remobilization of the silica in those grains, but must have had an exogenous source of silica. And when we combine this trace element evidence with petrographic indicators that these cements formed at a pre-compactional stage, as well as our paleontological observations, most parsimoniously, this suggests that ultimately the source of that silica may have been seawater, may have been silica-enriched bottom waters. But what about some of those other examples of Ediacara-style preservation that I alluded to previously? What about, for instance, some of our younger instances of Ediacara-style preservation from the other side of the Ediacaran-Cambrian boundary? So in order to tackle the question of whether or not other examples and younger examples of Ediacara-style preservation follow similar pathways of fossilization to that we see in the Ediacara member, I turned to the Elk Mound Group of Central Wisconsin, which is actually a unit that I was privileged to work on as an undergraduate student at Amherst College. And the Elk Mound Group is middle to upper Cambrian in age. Like the Ediacara member, it's a sandstone-dominated succession. However, the facies in the Elk Mound Group have long been used to infer a shoreline paleoenvironment. And in fact, this is a unit that contains trace fossils of some of the earliest animal forays onto land during low tide, and also importantly, contains a number of Ediacara-style body fossil assemblages, not only moldically preserved arthropods of a variety of taxonomic affinities, but also scyphozoan medusae or jellyfish. And you can see one of the more phenomenally preserved examples here in the upper right, where we actually can see the sort of lappets of the bell margin, the manubria, portions of the oral arms. And here on the left-hand side, you can actually see a dense assemblage of these scyphozoan medusae that have been interpreted as stranding events on the Cambrian shoreline. So if we apply the same tools that we use to characterize fossilization in the Ediacara member to these Ediacara-style fossils of the Elk Mound Group, what do we see? What we see at first glance, if we characterize, for instance, some of these scyphozoan medusae, is that these too are very silica-rich rocks. In fact, the Elk Mound Group fossils are orthoquartzites. They are quartz-cemented quartz aeronites, in which, much as we saw in the Ediacara member, these quartz cements include the entire porosity of these rocks, excluding all their phases, and occur as syntaxial overgrowths around the detrital quartz grains. And when we use laser ablation to measure the trace element composition of these cements, and what you see here in these white lines are actually scars from that laser ablation work, what we see is very much in keeping with what we saw in the Ediacara member, and that the quartz cements associated with these fossils are characterized by significantly different and slightly higher germanium silicon ratios than what we see in the surrounding detrital quartz grains here in gray, indicating again that they cannot have formed through remobilization of the silica in those grains, but must have had an exogenous and likely ultimately a seawater source. So if we begin to pull together these observations for both Ediacaran and Cambrian examples of Ediacara-style fossilization, the series of events that we might reconstruct is that the death and the rapid burial of Ediacara-style communities, both macroorganisms and the backgrounds that they inhabited, were relatively rapidly followed by the precipitation of diagenetic silica cements from upper pore waters that were still connected to overlying silica-enriched ocean waters. And it's likely that the organic substrates provided by these buried macroorganisms and buried macrounds would have served as sites for nucleation for those silica cements, as well as precipitation of cements in the surrounding pore spaces. And this early initiating and then continuous mode of silicification would have provided the glue, the scaffolding to hold together the sands grains, draping those buried organisms. And critically, that would have happened prior to significant tissue degradation and collapse, promoting the preservation of all of this extraordinary anatomical detail. And early silica cementation also offers some explanation for some of the more unusual sedimentological features we see in units like the Ediacara member and the Elk Mound Group, such as the sandstone-hosted sandstone interclass. So early silica cementation provides us with a mechanism for that early lithification required in order to create these clasts and resediment them in this manner, as well as for their preservation as discrete sedimentary and diagenetic features within these sandstone bedding planes. So more recently, we've been attempting to corroborate these observations that we've made from the Ediacaran and the Cambrian fossil and sedimentary records with an experimental approach. And this is work that's being led by my PhD student, Sylvina Slaughter. And what Silvina has been doing has been immersing various sorts of organisms, including invertebrate animals that are of course soft tissued, including sponges and anemones and cnidarians, in silica enriched artificial seawater with an initial composition of 2.2 millimolar. So initially at saturation with amorphous silica, and also maintaining a normal marine pH throughout the duration of these experiments. And what we've observed is that a wide range of these different organisms appear to undergo silicification on very rapid timescales, on the order of hours to days after the initiation of these experiments. And this silicification manifests itself as the formation of these white crusts or veneers that you can see in panel B and C here, which in electron microscope view consist of nanospheres of amorphous silica. But what we found particularly fascinating is that this silicification can occur and can continue even after the dissolved silica levels of the ambient experimental solutions have dropped well below 2.2 millimolar. In other words, even after the solution chemistry has dropped well below the thresholds that from a thermodynamic and kinetic perspective, we would typically consider necessary to achieve saturation with amorphous silica and induce silicification. So in order to dig into why it might be that silicification can occur even when solution conditions are undersaturated, Silvina, along with our colleagues at University of Alberta, Vedu Hao and Kurt Kaunhauser, also performed potentiometric titration analyses in which they characterized the reactivity of the tissues of the organisms that we used in these experiments, and in particular, the reactivity of organic functional groups that might be amenable to silicification. And what we found is that a wide range of the organisms we used in our study are characterized by the presence of organic functional groups that are particularly prone to silica absorption. And these include carboxyl groups, as well as phosphoryl groups, and hydroxyl and amino groups. And what we've observed is that the abundance of these organic functional groups, the density of reactive sites, as you see here on the x-axis, appears to be strongly and positively correlated with extent of silicification, as you see here on the y-axis, normalized to mass. And a wide range of the organisms we considered fall along this curve, suggesting that a wide range of different organisms might be amenable to fossilization by means of this pathway, but also addressing our question of how could silicification proceed when solutions are undersaturated by it is the organisms themselves and the presence of these silica reactive functional groups that is driving silicification. More recently, Silvina has been expanding the scope of these experiments to include a wider range of organisms, not only a variety of groups of animals, but also a number of different groups of microorganisms, including additional cyanobacteria, as well as representatives of red, green, and brown microalgal groups. And what we found thus far is very much in keeping with what we observed in our earlier study, that they appear to also be characterized by a strong positive relationship between the density of silica reactive sites of those silica reactive organic functional groups and extents of silicification. But what I find particularly striking here is that these microorganisms are characterized by some of the highest reactivities and the highest extents of silicification that we have yet measured. And in some sense, this is perhaps not surprising. In fact, there's a rich body of literature for cyanobacteria in particular, indicating that a number of species of cyanos are particularly prone to rapid silicification in silica-rich environments like sinters and hot springs today. But when we consider that these various bacterial and algal microorganisms are perhaps among some of our best candidates for what sorts of organisms may have populated Ediacara backgrounds, I think the ramifications of these experimental data are really quite exciting, and that they suggest that a role for Ediacara backgrounds that was not only linked to supporting the ecology of Ediacara organisms, but also their fossilization. And this would suggest that where these backgrounds were present and where they were well developed, they would have amplified the fossilization potential, the silicification potential of surrounding Ediacara macroorganisms living on and in them. So if we begin to pull together some of these various observations from both fossil and sedimentary archives and our experimental data, early silica cementation offers a compelling explanation for many instances of Ediacara-style fossilization. And moreover, given its link to global marine biogeochemical conditions, so the high dissolved silica levels characteristic of the Ediacaran Ocean, it's likely that this would have operated as there would have been a global scale potential for fossilization by means of the early silica cementation pathway, regardless of local differences in facies or fauna. Moreover, both our experimental data and our paleontological observations suggest that a wide range of different sorts of organisms, of different body plans, of different tissue consistencies were amenable to fossilization by means of this pathway. And in that light, it's quite likely that Ediacara-style fossil assemblages are not unduly biased, but do hold the potential to provide us with faithful archives of Ediacaran and early Paleozoic seafloor communities, which is something that perhaps has been previously assumed rather than directly demonstrated. Moreover, given that the stratigraphic record of the Ediacara-style fossil assemblages extends all the way up through the lower Paleozoic, and given that with this more detailed scrutiny of Ediacara-style preservation, we see evidence of the same early silica cementation pathway operating in both Ediacaran-aged and Cambrian-aged Ediacara-style fossil deposits, I think that this robustly demonstrates that the disappearance of the Ediacara Biota at the close of the Precambrian is unlikely to be a preservational artifact, but is more likely to be a real evolutionary signal. And finally, I think it might be no coincidence that the timing in which we see the eventual closure of the Ediacara-style fossilization window toward the close of the lower Paleozoic is the same as that in which independent geochemical and sedimentological and paleontological indicators suggest was a time of major radiation among our silica biomineralizing groups, particularly our silicifying zooplankton, the radiolarians, which appear to increase greatly in both diversity and abundance at this time. So it may be that the closure of the Ediacara-style taphonomic window is ultimately the signal for the advent of a more biologically regulated modern style silica cycle. And now that we have a better handle on some of the mechanisms that are responsible for Ediacara-style fossilization, I think that we can finally revisit some of those larger scale questions with which I began the talk, such as perhaps most pressingly, whatever happened to the Ediacara Biota? Given that, as I think these data robustly demonstrate, declines in Ediacara taxonomic diversity in the latest Ediacaran and the eventual disappearance of the Ediacara Biota at the very end of the Ediacaran are unlikely to be preservational artifacts and thus therefore be real signals of evolutionary extinction. So what factors might potentially have driven the Ediacara Biota to extinction? And with all things related to the Ediacara Biota, there have of course been no shortage of ideas related to this question of Ediacaran extinction, but many of these ideas fall under the larger umbrella of environmental perturbation, and in particular, the potentially potent combination of carbon cycle and redox perturbation. So it's upon that particular model that I'm going to be focusing for the last few minutes of my talk. And of course, the Neoproterozoic stratigraphic record has long been characterized by hallmarks of extreme carbon cycle perturbation in the form of some of these really dramatic negative carbon isotope excursions. Perhaps most famously, the Shurham negative carbon isotope excursion, which really stands out in terms of both its severity, its magnitude that is, and its duration, but is also accompanied by both older and younger instances of extreme carbon cycle perturbation, including the basal Cambrian negative carbon isotope excursion, or BASE, which has been suggested in a number of successions worldwide to potentially correlate with the Ediacaran-Cambrian boundary. And of course, these negative carbon isotope excursions are themselves a question of considerable debate. We could spend not only a whole talk, but many talks discussing all of the various models that have been put forward. But one of the ideas that perhaps has received some of the greatest attention is the idea that the extremity of these excursions requires oxidation of a large reservoir of isotopically depleted organic carbon. And the idea has been that the entire reservoir of particulate organic carbon in the Precambrian oceans would not have been sufficient. And there must have been another larger reservoir of isotopically depleted carbon to be oxidized to account for these signals. So from that idea, there has in turn been a suggestion that the Precambrian oceans would have been characterized by an extremely large reservoir of dissolved organic carbon, or DOC, and that the episodic oxidation of that large DOC reservoir must be the factor that principally explains these negative carbon isotope excursions. And a couple of other accompanying ideas that have led to the emergence of the DOC hypothesis have been the observation that many neoproterozoic successions are characterized by geochemical evidence for low background dissolved oxygen levels. So it's been suggested that the oxidation kinetics that would be required to break down this large DOC reservoir may have been somewhat more sluggish. And others have suggested, foremost Nick Butterfield, and from his work has emerged what's been called the Butterfield Ocean hypothesis, that given that Precambrian primary producers in the surface ocean were predominantly cyanobacteria, and given that cyanobacteria have on average smaller size cells than do their eukaryotic primary producer counterparts, that the idea has been that upon the death of those communities, as their cells began to sink through the water column on their long journey toward the sea floor, that they would have sunk more slowly than would larger eukaryotic cells, and thus may have been exposed to broader opportunities for degradation to DOC and remineralization. So from this sort of coupled observations of the carbon isotope record, of low background dissolved oxygen levels, and the ideas that have gone into the Butterfield Ocean, it's been suggested the Precambrian may have had a truly enormous dissolved organic carbon reservoir, up to 30 times higher than the inorganic dissolved carbon reservoir that we see in the modern oceans. And this idea of a large DOC reservoir has so taken a hold of the community view of the Ediacaran and of the Ediacaran biosphere that it's been suggested that many different Ediacara Biota taxa may have been particularly adapted to life in this organic rich, soupy, high DOC ocean conditions, and that they may have even practiced osmotrophy. This has been particularly suggested for some of our rangiomorphs, our fractal frondomorph fossils that are particularly predominant in the Avalon and some of our older Ediacaran fossil assemblages. So these organisms have been proposed to have fed themselves by means of direct uptake of dissolved organic carbons across their external tissues or cellular membranes. And thus the catastrophic oxidation of this large DOC reservoir may have been commensurately catastrophic for Ediacara Biota organisms, either by directly jeopardizing their survival or making them ecologically less competitive relative to members of burgeoning, more Phanerozoic style animal groups emerging at this time. However, the very idea that Ediacara organisms would have been susceptible to the oxidation of a large DOC reservoir, and in fact, the very existence of such a gigantic DOC reservoir in the Precambrian oceans have been rather more areas of supposition than things that have been directly and empirically demonstrated on the basis of understanding of the mechanisms that regulate carbon cycling in the oceans today. So in order to address these questions, and more specifically to address the question of whether or not the oxidation of a large DOC reservoir could offer some explanation for Ediacaran extinction, my colleagues and I recently developed a new ocean biogeochemical model coupled with a Lagrangian particle aggregation disaggregation model. So in a nutshell, this combined model encompasses a range of processes that we observe in the modern oceans to regulate cycling of both inorganic and organic carbon in the oceans, from productivity across a variety of planktonic groups and size classes, to particle aggregation, whether through biogenic or lithogenic ballasting, or through fecal pellet production, or through the inherent stickiness of organic matter, to disaggregation, whether through degradation of particulate organic carbon, or through the activities, for instance, of coprophagous zooplankton. And this model also includes a reactive transport expression of how carbon is cycled in the water column during production and export productivity by means of a variety of microbially mediated respiration pathways. So in other words, the processes that dictate how particulate organic carbon is converted to dissolved organic carbon, which is in turn converted to dissolved inorganic carbon. And this model also considers environmental boundary conditions that have been strongly linked to carbon cycling, such as temperature and dissolved oxygen concentrations. So having assembled this model, we of course wanted to convince ourselves that it was a reasonable facsimile for observations that we would make in the modern world. So we used this model to determine if we could reproduce modern DOC profiles. And what we found is that we could indeed, without need for any additional parameterization, closely replicate DOC profiles in all of the major ocean basins, as well as for lake systems, which today have served as important analogs for potential ferruginous and euxinic ocean behavior in the Precambrian. So having convinced ourselves that this model could indeed closely replicate real-world observations, we then performed a variety of sensitivity analyses to try to determine what sorts of combinations could feasibly lead to the development of a large DOC reservoir, or whether we could even reproduce the very large DOC reservoir that has been proposed to characterize the Precambrian oceans. And what we found is that even under some of the most extreme end member conditions that have been proposed for the Precambrian, such as the lowest boundary estimates for dissolved oxygen levels, or for instance, the very cold seawater temperatures that might have characterized cryogenian icehouse oceans, even under all of these more extreme scenarios, we could not reproduce the very high DOC reservoir that has been proposed to characterize the Precambrian oceans. And in fact, even with some of our strongest, what emerged as our strongest levers upon this system, such as seawater temperature, we observed only modest inflections around the values that we observe in modern ocean basins. And ultimately, this is due in part to not only what other authors have previously highlighted under the dilution hypothesis, that although DOC, when it's very diffuse, may not be an appropriate substrate for microbial respiration, but when it's concentrated, it will very rapidly become respired and remineralized, but also more fundamentally because the rates of production of dissolved inorganic carbon from dissolved organic carbon essentially keep pace under all of these scenarios with the production of dissolved organic carbon from particulate organic carbon. So in this slide, the answer to the question of whether the oxidation of a large DOC reservoir could feasibly explain Ediacaran extinction is no. However, of course, this doesn't necessarily close the door upon the importance of other environmental factors in influencing Ediacaran extinction. And my hope is that future work, and in particular, improved chronostratigraphic constraints, allowing us to tie together observations from different assemblages worldwide, as well as a greater number of studies that collect paleontological and biogeochemical data in tandem, will allow us to return to and reinterrogate the question of whether Ediacaran extinctions on the whole followed patterns characteristic of Phanerozoic mass extinctions, in which the rise and the fall of evolutionary faunas and the emergence of ecological novelties has been punctuated by episodes of extreme environmental perturbation. And with that, I'd like to thank a number of colleagues who were instrumental to this work, as well as various sources of funding for their support, and of course, all of you for your attention. And again, Andre and Alex for organizing this seminar series. And I'm happy to take any questions.
That was great, Lydia. Thank you so much for that. We're going to transition to a Q&A session, which you can type into the chat or you can raise your hand virtually. I posted in the chat a way how to raise your hand for people who weren't sure. We have a couple things already in the chat. I see Margosha is there as well. But Paul really quickly mentioned one problem with the Butterfield Ocean is that fine-grained particles in seawater tend to flocculate and sink as larger particles aggregate. And do you have a comment for that?
Yeah, I would entirely agree. And that was one reason why I think it was important that we included some of those aggregation processes in our modeling approach. So yeah, organic matter is inherently very sticky and tends to aggregate exactly as you've highlighted, even without the effect of ballasting or fecal pellets. So I think that certainly is a mechanism that that would suggest that looking to cell size alone is perhaps not an adequate proxy for what we expect export productivity to have been like in Earth's past oceans.
Right, and I wondered from that question if silica precipitation on those organic surfaces could have impacted this flocculation, and it's, because you were mentioning that it can happen on the scale of days, correct?
Yeah, so that's what we observed in our taphonomic experiments, certainly, is that it can happen very rapidly. And that's something that has, in terms of your question about whether or not this is a process that could happen in the water column, this is something that has been previously proposed. I think some of the work by Berger Rasmussen and others has suggested that potentially, at least in, say, the Archean, when we think that dissolved silica levels may have been particularly high, maybe you could actually have silicification occurring in the water column. And there's, of course, been a lot of debate about that in the context of, for instance, banded iron formations. And I think that's a really intriguing question. And of course, if that did occur, we might expect that too to influence export productivity.
Okay, we have one from, thank you. We have one from Eric Sperling and then Margosha. Hi Lydia, great talk. In a recent paper led by Lizzie Trower, Geobiology 2021, we suggested that seawater silicon concentrations were relatively low by the middle to late Ordovician. I was wondering how you saw the evolution of seawater silicon concentrations through the Ediacaran through Paleozoic. Does this existence of fossilized medusae in Upper Cambrian sandstones with similar preservation to Ediacaran organisms, but relatively few in Ordovician, to my knowledge, potentially suggest a change around those times? You talked about this a bit. I would appreciate hearing more.
Yeah, that's a really great question. And I think that one that has been very usefully highlighted by your and Lizzie's recent paper in Geobiology. I think it's still a little bit murky for me, you know, what happens to dissolved silica levels as we cross that transition. I think the existence of Ediacara-style fossil assemblages like the Elk Mound Group do suggest that dissolved silica levels were still higher in the middle to upper Cambrian, but exactly when they began to decline, whether or not dissolved silica levels remained relatively high before the emergence of diatoms, for instance, and if that was the final blow, or to what extent radiolarians or silicifying sponges, for that matter, which although not as tend to be not as dominant in terms of their environmental scope as, say, planktonic groups, but whether or not they could have also played a role in driving down dissolved silica levels. I think one of the things that's intriguing is not only the presence, although I agree they are less abundant in the lower Paleozoic than what we see in the Neoproterozoic, of Ediacara-style fossil deposits like the Elk Mound Group, where the youngest instance of which is, I believe, the Devonian, but it's sort of an outlier there. But also some of the older arguments from sedimentological archives, for instance, of things like silicified oolites in the Cambrian and the Ordovician, as well as shifting locus of chert deposition, as folks like Woody Fisher and Andy Knoll and of course and Maliva have their work has long highlighted. That would suggest that the early Paleozoic is a very interesting time in that regard. So my suspicion would be that dissolved silica levels don't immediately drop as we see the emergence of our earliest silica biomineralizers like silicifying sponges. But I think it's not improbable that they drop to some extent, whether it's as to greater an extent as you and Lizzie recently proposed. I think there's still some debate to be had as to that. But I think that it's, one of the pivotal questions for understanding ancient silica cycling is where were the inflection points and how steep were they? And of course, there's the question of not only diversity of silica biomineralizing taxa, but also their ecological and environmental abundance.
All right, thank you. Margosha, would you like to talk?
Yes,
or ask a question?
Thank you. It was a great interpretation and a lot of work. I appreciate it very much. But why do not we have preserved organic matter in any of the Ediacaran fossils, with the exception perhaps one occasion reported by Ilya Bobrowski? Why don't we have organic tissue preserved? At the same time, in similar rocks, we have preservation of organic microfossils. why not large Ediacaran type fossils?
And that's also a great question. I think, a partial answer to that is that, if we were to look to sort of analogous environments today, sort of sandstone dominated or sand dominated environmental settings tend to be, or the fanners for that matter, don't tend to be characterized by extensive organic matter preservation. And that's one reason I think why, sandstone successions that are shallow marine or intertidal have long been known for their phenomenal trace fossil records, for instance, but not typically for their phenomenal body fossil records. Whereas throughout the geologic timescale, and in particularly in certain intervals of Earth's history, like the Neoproterozoic and the lower Paleozoic, we see really phenomenal preservation of organic matter in muddier sediments. And I think there are, of course, a number of sort of models that have been put forth from the work of Bob Gaines or from Ross Anderson and Nick Butterfield and others suggesting what could enhance preservation of that organic matter and the potential role of clays or orthogenic carbonate cements or other factors. And we could spend all day just talking about those really interesting windows of fossilization. But I think that, I wouldn't tend to expect that sort of sandstone dominated coarse grain deposits would be characterized by really extensive organic matter preservation. And the other thing that I can say is that on the experimental side, what Silvina has been observing, though I didn't have time to go into it in more detail during my talk, is that the formation of these silica crusts, and also recently she's been observing the formation of silica cements, and some of the more recent experiments she's been doing with sandy substrates are also associated with total degradation of organic matter by the end of the experiment. So the TOC by the end of these experiments is also essentially null. Yet we nonetheless see the formation of these silica precipitates and these silica molds. So I think sort of what's critical there is, especially in these sort of coarser and more porous sediments in these higher energy environments that are represented by many of these sandstone Ediacara-style units, is whether or not silicification can occur quickly enough to outpace degradation. Degradation is still occurring. I mean, degradation is occurring in many of our windows of exceptional fossilization, and in some cases is integral to their preservation. And it may be that degradation is necessary to some extent in order for these organic functional groups to react with dissolved silica, with silicic acid. But I think that I wouldn't expect that really that the silicification could occur quite so rapidly as to totally encapsulate the entirety of these macroorganisms, some of which are over a meter in length. But I think what could be critical is that you initiate the formation of these silica cements. And it really doesn't take all that much cement to sort of keep that scaffolding intact of those sand layers burying those organisms. And that might be enough, especially when it's added to during the later stages of early diagenesis and mesodiagenesis to facilitate the preservation of these molds. But it's a very good question.
Thank you.
Okay, we have a couple more in the chat. First from Daniel Fitzgerald. Great talk. Have you considered how the functional groups which are driving the silicification on a rapid time scale would impact these organisms during life, i.e. some level of entombment preventing proper feeding?
Yeah, that's also a good question, and one that sort of follows a little bit on my response to Margosha's question. So I don't think that the dissolved silica levels are quite rapid enough that we would expect there to be sort of wholesale entombment and certification of these macroorganisms, particularly given what for many of them was a quite large size. So it's an interesting question of whether or not there could have been danger of silicification occurring during the lifetime of these organisms, such as has been observed, for instance, in some sinter environments for cyanobacteria today. So I suspect it's not quite high enough to drive that. And that sort of brings us more in lines with sort of the range of estimates for dissolved silica levels that have been proposed for the Ediacaran. And the importance of these functional groups and sort of overcoming those kinetic limitations to saturation is quite critical as well. But it may also be that some amount of initial degradation assists with the absorption of silica to the organic functional groups in the tissues of these organisms.
All right. Our next one is from Greg Vitalik. He says, nice job, Lydia. I agree that the pyrite death mask should be retired and silicification, best known in fossil plants, is more promising. But how do you explain the germanium silicon ratio is 3 to 10 times more than known in any aquatic system, marine or non-marine, and more like soil solutions?
So yeah, this is a question we've discussed before, Greg. And I would say, my first response would be that on average, the germanium silicon ratios in these cements is not that severely elevated above those of the surrounding grains. And for instance, if you saw the values from the Elk Mound Group fossils, the average, well, in both cases, both the Ediacaran and the Elk Mound Group, the average is something like 2 or 2.5 for germanium silicon ratios in those cements. So I wouldn't say that necessarily places us very far outside the range of what's been observed in aqueous solutions, including marine ones before. But more importantly, I think that this also exemplifies the importance of autogenic processes. Germanium is known to be an organophile. So we would expect that drawdown of germanium from those silica-enriched pore waters may have been elevated even relative to drawdown of silicon during the early diagenesis of these assemblages. And therefore, we might expect that there is some sort of fractionation occurring such that the cements are characterized by higher germanium silicon values due to the organophile tendencies of germanium relative to what the surrounding pore waters would have been characterized by.
Okay, we have another from Margosha.
No, this was the repetition. Sorry.
Oh, go ahead. Go ahead.
Yeah. There is one, next one. May I?
Yeah.
Okay, Lydia, I like very much the interpretation, and of course, based on good evidence, of the higher content of silica dissolved in the Precambrian waters. And this was one of my, supportive arguments to interpret those morphotypes of metazoans from northern Norway, which have siliceous skeleton, exoskeleton. And I don't know if you have seen it, and would you agree on the interpretation? Those are marked.
Go ahead, sorry, I didn't mean to cut you off.
Very much below the Ediacara type fossils in the same succession. And have you seen those fossils, or I mean the publication?
Yeah, I am familiar with your publication. And yeah, it's very intriguing material. I think that, first of all, I think that, these are the sorts of things that should be within our search image, is not only sort of classic Ediacara-style and Ediacara-type macrofossils, as well as things like textured organic surfaces, but also in perhaps, more enigmatic features that may have previously escaped detection and might be telling us something about marine biogeochemistry, including dissolved silica levels. I think one of the challenges for, many of these silicified deposits is trying to distinguish between sort of primary and autogenic processes. Because if I recall properly, and correct me if I'm wrong here, one of the interpretations that that you and your co-authors laid out was that perhaps this was evidence for actually biomineralization. Is that right?
Yes, absolutely.
Yeah. So, and it's a really important question of why don't we see more instances of silica biomineralization in the Ediacaran? And it's long been tied up with some of the debate about the sponge fossil record, for instance. As Eric once said in one of his papers, where's the glass, right? If we had high dissolved silica levels, why don't we see silicified sponge spicules from earlier on than the lower Cambrian, which appears to most recently be sort of our oldest endpoint for well-preserved spicules. So I think some of the work that Shuhai and his colleagues have done sort of suggesting that maybe some of our early spicules had sort of an organic template is really quite intriguing. And I think it also goes hand in hand with broader questions of, why not carbonate biomineralization earlier than what we see it in the Neoproterozoic fossil record as well. So I think it's it's long been a perplexing question of, do we see evidence of silica biomineralization, or why don't we see more instances of it earlier in the Precambrian, given that dissolved silica levels were so high? But I think also one of the big challenges moving forward is going to be to distinct, and this is a challenge that has sort of equally plagued attempts to identify carbonate biomineralization or apatite biomineralization, as how can we develop tools that allow us to sort of reliably distinguish between mineralization during the lifetime of the organism and mineralization that may have happened during very early diagenesis. So I think it is an exciting arena for future work as well.
Thank you.
All right, Shuhai has his hand up.
Yes, thanks, Lydia. This is a great talk. So I have a couple questions, very quick ones. The first one has to do with your iron oxide and the uranium disequilibrium work. I think I wouldn't be surprised that the iron oxide will precipitate very recently. But my question is, how can you use this data to falsify by itself? I'm not saying that this falsification hypothesis was incorrect or anything like that, or just this line of evidence by itself does not imply that there was a precursor, there was pyrite that were present. In fact, the pyrite death mark hypothesis. So yes, I don't know if I make myself clear enough. So basically I'm saying, yes, the iron oxide was late, but there may be pyrite precursors that were precipitated in the Ediacaran time.
Yeah, that's a great question, Shuhai, and one that I've wrestled with as well. And I think, ultimately the answer is that we can't be entirely certain. We can't entirely rule out that there may have previously been pyrite on some of those surfaces, though we don't see petrographic evidence for it. We don't even see that iron oxides within these fossils are common components. Again, in the Ediacara, remember, it's really quartz cemented, quartz feldspar aeronites with not really any other phases except these coatings along these bedding planes. And that includes when we have intrastatal preservation of fossils as well. But I think, ultimately I think the utility of things like the sedimentological characterization of these iron oxides as having classic facies that we would associate with reaction fronts, as well as the uranium isotope evidence, what that really drives home for me is that, sure, we could speculate that there may have been pyrite previously on these surfaces, but when the geochemical indicators tell us that what those phases that are present have been open systems, we really can't use them to say anything either for or against the death mask model. In other words, like if our geochemical indicators fail the very basic test of whether or not these can tell us something about syngenetic conditions on these surfaces, I think that we really need to sort of leave those lines of evidence to rest and no longer invoke them in favor of a particular hypothesis, if that makes sense. So although there's sort of been a lot of inertia, a lot of sort of intellectual development that has gone behind the idea of the death mask model in the decades since it was first proposed, I think if ultimately we find that the principal line of evidence in favor of the death mask, which was the presence of those oxides, is something that we can't reliably use to say anything about geochemical or sedimentological or fossilization conditions, we have to sort of leave that as, there's absence of any evidence suggesting the death mask model, even if at the end of the day, we might surmise or speculate that maybe there was an entire untold history of pyrite formation much earlier in the history of these successions that has since been wiped away. If it's been wiped away, we really can't invoke it as evidence for anything related to fossilization or ocean biogeochemistry would be my perspective. But I think on the whole, the uranium isotope evidence we do see is suggestive of actual precipitation of those oxides under oxidizing conditions, rather than oxidation of pyrite deposits that may have been in place at some point in the earlier history of these sediments. And we commonly observe features like Lisa-Gang banding, and again, these very abrupt reaction fronts that I think should really give us caution in terms of assigning sort of any sort of process of Ediacaran age to the formation of either those oxides or their precursor phases.
Okay, well, fair enough. My other question is follow up to Margosia's question. So I was a little amazed, I was intrigued by the similarity and the differences between silicification in the Dershan Tor, for example, and the silicification in the Ediacaran mantle. There are certainly some similarities, but I was intrigued by the differences, and that's in the Ediacaran mantle, you don't have any microfossils, whereas in the Dershan Tor formation, so only microfossils. And they're clearly microorganisms in the Ediacaran mantles. You can see this microbial mats and things like that. So why these microbes were not silicified, whereas the macroorganisms were?
Yeah, that's also a really good question. And I think, that one of the things that we have to keep in mind is that the grain size of the Ediacaran member is such that we wouldn't expect microorganisms to be sort of very readily preserved in that matrix as molds and casts, because we're looking at sort of fine to coarse grain sand. So I would say that the grain size alone is sort of inhibiting of everything except things that are macrofossil grade, maybe on the very small end of macrofossils. Like for instance, it took years before we found a candidate tracemaker body fossils for instance, the helminthoid ignites. And I think that's in large part because of the grain size inhibition on really moldically preserving microfossils at the cellular level. So I think that's something that we have to bear in mind. And when we do find finer grain deposits, such as were critical to recognition of that tracemaker, which was something that was myofaunal scale or very small macrofaunal scale, it can be really sort of eye-opening. But in many of our Ediacara member deposits, I think the grain size is simply too coarse for us to expect that. And then I think the other thing is sort of, going back to Margosia's comment that we don't really see sort of certification of sandstones all that regularly. At least I think, this is something that we characteristically see either in sort of carbonate hosted deposits or in muddy or silty deposits, but it's not something we see at even other times in the Precambrian fossil record as being very characteristic of sandstones. So I think this means that although high dissolved silica levels can contribute to various modes, various windows of exceptional fossilization, whether it's silica replacement, such as we see, for instance, in some of our Phanerozoic shelly fossil deposits, or permineralization, or microfossil preservation in cherts, or Ediacara-style moldic fossil, or even perhaps a role played by high dissolved silica levels in orthogenic clay formation. These can operate as very different taphonomic modes under the influence of not only different taphonomic conditions and different faunas, but also different lithologies and different facies.
Mm. Yeah, the overgrowth rim seems to be 10, 15 micron. So I'm not, the grain size is big, but the precipitates is big enough to entomb some of the microbes. If they're 10 micron or small, they should be preserved, but I don't know why. There might be some taxonomic preference in this taphonomic window.
Yeah, no, it would be interesting. And I don't know if it might be that it's a question of scale, and it may be that your sort of initial silicification is sparse enough that you can capture some of the salient morphological details for your macroorganisms, but not extensive enough to capture all of the sort of necessary morphological information that would be required to identify molds of bacteria or other microbial grade organisms within those cements. Or, you know, it might be that as we keep looking, we'll eventually find molds of those microorganisms within the thicker rims that we see in these cemented sandstones. So certainly it's something we're keeping an eye out for. But I also think that there are sort of compelling sedimentary and facies reasons, as well as sort of what we envision to have been the sort of uneven rate of silicification throughout the diagenetic history of these assemblages that could contribute to why we haven't yet found molds of those microbial grade organisms in our cements.
Okay, great. Thank you very much.
Great. Daryl, you have your hand up next.
Yeah, a quick question. I'm intrigued by this early silicification of the seafloor producing intraclast layers. Do they have any specific shape? Are they rounded at the edges? Do they taper towards the inwards from the edges? Is there any criteria that you could see, or are they just blocks?
Yeah, they're blocks of varying sizes. I would say that texturally they range from subangular to subrounded. And they're of variable shapes as well. Some of them they're not commonly, you know, sort of, very, very spherical or very round. They're sort of tabular. They're sort of reminiscent of flat pebble conglomerates, for instance, that we might, you know, characterize from lower Paleozoic successions, except that they're sandstones and we don't see evidence of carbonate cement. But they are subangular to subrounded, commonly, and they're tabular, so they're not very thick three-dimensionally, and they're very common along the upper surfaces of these sandstone bedding planes, which is also quite intriguing and sort of not what we would expect for, you know, sort of cobbles, for instance, that would be reworked and might, you know, be a lag at the base of a tempesta, at the base of an event bed. And we see sort of varying degrees of disaggregation of the surfaces, and which is why your sort of term block was in some ways quite apt. Like we see everything from surfaces in which are sort of just at the initial stages of breaking up and ripping up, where we just sort of see, you know, sand cracks and sort of the pulling away of pieces of what was presumably like an incipient rip-up surface, all the way through to relatively smaller sized, you know, sort of very coarse to granule sized class that are sitting within a sandy matrix that doesn't look anything like the mat ground, and our sort of indicator that they may have been derived from a mat ground comes from not only that brittle behavior, but also occasionally we do see textured organic surfaces on the individual class that have been redeposited in those beds. So that's sort of those are the, you know, the primary lines of evidence that have suggested to us that they might be derived from mat grounds, and that those mat grounds must have undergone early lithification, what seems to be through silica cementation. And we observe them only in the Ediacara. Remember, we observe them in particular facies as well that are characterized by, in some cases, one of our facies is characterized by evidence for transport and in fact as full of entirely transported fossil assemblages. And another facies in which they are more common is a little bit more of a distal, so we're upper incised valley infill facies, below what we interpret to have been below storm wave base, but still within range of sort of episodic disturbance, and we find tool mark surfaces and things like that. But interestingly, similar features have been described from other Ediacaran and Cambrian age sandstone dominated successions. And in fact, Pfluger and Gressa described in, I think it was in 1996, very similar features in the Ediacaran Cambrian Nudaus of Namibia. And they've also been described from the Elk Mound Group, which was that Cambrian aged unit in which I walk through some of the salient features that suggest Ediacara-style fossil assemblages, and those deposits are preserved through similar pathways. So they've perhaps not received as much notice as maybe they should, but they're a little bit more widely spread than I think has been broadly appreciated.
Okay, thanks very much. Great talk.
Thank you.
Hi, Lydia. Great talk. I have a sort of 3 comments, questions. First, sort of following on Shuhei, I have very similar comment about iron oxides. And so since you answered to him, I'm wondering if you go to avalon facies, which are deep water and more likely to have pyrite, do you see similar style of preservation? Do you argue again for absence of pyrite, or it's somewhat different from this shallow water facies where clearly like we're oxic and probably shouldn't be pyrite? So that's first question. Second question about a germanium silica ratio, and I'm wondering, like, since you have dissolution and when you have precipitation, wouldn't you expect anyway to have different germanium silicon ratio from quartz that may be formed in high temperature igneous conditions? And I'm wondering that probably a good test would be to go to younger sandstones that after biological silica secretion already started, and you probably can test if this quartz overgrowth on quartz grains that clearly are not related to silica precipitation from seawater, if they would have the same germanium silicon ratio as a quartz cause or not. And the third, just a comment on in terms of preservation of organic matter. I think one of the best places kind of to address why supposedly you preserve Ediacaran fauna by silicification and you don't have microfossil silicified might be on East European platform in Baltica, where like in Moscow scenic cliffs, for example, you have Ediacaran fauna and you have well-described benthic mats with rich microfossils fauna, but I think it's still not silicified. It's just preserved as organic world. So kind of 3 questions, comments if you have.
Yeah, no, those are all great questions. I think I'll go in reverse order, lest I forget the question. So the first is, yeah, that's a really interesting suggestion. I've actually really long wanted to do some work in the Eastern European platform. So perhaps that's further impetus to try to characterize some of the fossils, or even just some of the petrogenesis and the early diagenesis of those successions. So yeah, that would be a really sort of fabulous and an obvious follow-up to some of these major questions. The second question was about germanium silicon ratios. And I think it's not that we would expect that the value had to be the same as what we see in the detrital quartz grains, which have like sort of a classic continental signal, but rather that there's not an easy explanation for how we would go higher than those grains. If they are derived from the grains, we would expect them to be the same value or lower. But the fact that we observe that the value is higher is, I think, a really nail in the coffin for, in addition to the petrographic evidence indicating sort of lack of evidence for significant dissolution and reprecipitation, is that they weren't derived from those grains. So in terms of sort of pinpointing more specific mechanisms for why we might get that range of germanium silicon values, I think really sort of the most intriguing one, and the one in which we're most closely investigating now, is the potential sort of organophilic behavior of germanium, which may be even more elevated than that of silicon. And actually, Silvina, the PhD student's work that I presented, is currently doing some experiments exploring changes in germanium silicon in conjunction with these decay experiments. So we're hoping to get a little bit more of a ground truthing of the utility of germanium silicon and how much that varies across, for instance, modern taxa and dissolved silica levels and the like in the near future. But that's certainly something that's on our agenda. And then I believe the the final question, the first of your questions, concerned what we might expect in terms of pyritization in deeper water successions, such as those of Newfoundland and of the Avalon Peninsula. And Alex Liu, for instance, has published some work suggesting that pyritization might have played a more important role there. From what I've seen in terms of his SEM images, as well as his petrographic photomicrographs, there is pyrite within those sediments. There are framboids, but to my eyes, at least, they look pretty isolated. They don't really seem to be very strongly associated with the fossils and not with the matrix, and they're still relatively low in abundance. So they appear to be more abundant than what we see in the Ediacara member, and they are indeed still preserved as framboids, but to my mind, there's not really a systematic association with the fossils in those deposits. And in speaking with some of the folks who have done more extensive work in those successions, such as people from the British Geological Survey, I've also been told that some of those surfaces contain a range of sort of heavy metals that would be much more easily reconciled with recent fluid flow than they would with necessarily Ediacaran syngenetic pyrite formation. So I think there's still some really intriguing questions to be addressed from other Ediacara-style fossil deposits, such as those of the Avalon Peninsula. The other thing that sort of sets the Avalon Peninsula material apart, and I should say also that I haven't worked on it extensively myself, though I currently have a postdoc who's characterizing some of the preservation in the Formose formation, which is part of that overall succession, is that the Avalon ranges from being sort of sandstone dominated to more heterolithic. So in some places, we actually see finer grained siliciclastics comprising an important component of these fossiliferous successions. But the other thing that the Avalon has sort of long been known for is its ash deposits. And in fact, many of the turbidites that appear to blanket some of these fossiliferous surfaces are enriched in ash, which could potentially provide another source of dissolved silica, in addition to what we might expect to have been ambient in Ediacaran bottom waters. So it may be that proximity to this more readily dissolvable source of silica in the form of these ash deposits, in addition to whatever might have been ambient in Ediacaran seawater, could also drive silicification of some of the moldic fossils that we see preserved along the Avalon Peninsula as well.
Thanks, Lydia. We can talk about Baltica at some point. So, but for silica, would you expect if it would be overgrowth, you can still have probably some organic components and life present in shallow burial environment that could also affect germanium silicon ratio? I think it would be really interesting to pick up some Cenozoic or Mesozoic sandstone and try overgrowths, and if you see something different from earlier overgrowths.
Yeah, no, I entirely agree. And sorry, I forgot to circle back to that part of your question. So that's actually something that we're trying to do right now in my lab using the laser ablation instrument that we've recently set up, and actually have a postdoc who's currently not only doing germanium silicon characterization of, right now we're doing principally Precambrian, but the idea has always been to also move into the Phanerozoic for exactly these sorts of issues, in order to not only find, sort of an outgroup upon which these sorts of proxies can be further ground truthed, as you're suggesting, but also sort of in pursuit of the questions that Eric raised about, what happens during the early Paleozoic, or what happens later in the Paleozoic? When do dissolved silica levels appear to really decrease? And the other thing that my postdoc Zhuan Wang has been doing is setting up silicon isotopes between our laser ablation instrument and our Neptune multi-collector. So we're hoping that will also sort of open more doors in terms of sorts of material and the sorts of questions that we could address using not only a trace element approach such as germanium silicon, but also the silicon isotope system.
Thank you.
All right, we've got one more from Greg over here in the chat. Aren't your red brushes important evidence for Ediacaran oxidation because red within white or gray matrix?
Yeah, so this is of course also a discussion that we've had a few times before, Greg. But I would say, you know, again that as I've said before, that the presence of red, of color, is, you know, an indicator of the current composition of those sediments. And in some cases that might be an original signal for iron 3, and in some cases it might be a signal for iron 2 that was subsequently oxidized. There also are sort of a wide range of reasons why you could see differences in color even within an individual sedimentary unit or even an individual bed, such as, you know, differences in grain size, differences in texture that might mean that phases are more or less prone to oxidation or reduction, even during the not only the early diagenetic history of those sediments or the sedimentary history of those sediments, but even during later stages of diagenesis or even, you know, tectonic and metamorphic activity later in the burial history of those assemblages. So I don't think that sort of differences in color really provide us with a very diagnostic proxy for whether or not, you know, whether or not what was sort of the original iron content of those sediments, which is something that's notoriously hard to track, even in across a wide range of geochemical proxies, and certainly not for allowing us to distinguish between marine and terrestrial environments.
All right. Well, you fielded all standing out inquiries. So, and you did very well. I really enjoyed this presentation. Anybody have any other comments, thoughts, questions? No? All right, I think we've taken an hour and a half of your time now. So you did a really good job. Thank you so much.
Thanks so much for the invitation to speak in this seminar series. I've really enjoyed the entire seminar series so far. So thanks again for that opportunity and for the great questions.
Thank you very much, and bye-bye.
Bye.
Yeah, take care. See you around.
Bye. Thanks.